Okay, so in this section, we're going to be looking at proteins and enzymes. And this follows on from the session about amino acids. So as we just said in the last session, amino acids are often called the building blocks of life. They're very like little plastic building bricks, but for chemistry, instead of putting them together to make houses and rocks, they're used to make all the proteins in your body. So it's why we're sort of going on from amino acids to proteins, because if we join all those amino acids together, as you know, you get proteins. And we're also going to look at enzymes, as enzymes are a form of proteins. So proteins are a form of condensation polymers of amino acids. So proteins are made up of lots of amino acids. And these amino acids have been joined together by peptide links. The chain is put together by condensation reactions, and then they are broken apart by hydrolysis. So very similar to when we talk about those polypeptide chains. So they're condensation polymers, not addition polymers, condensation polymers. So they're joined by condensation reactions, and then they can also be broken apart back to their monomers by hydrolysis. So as you can see at the bottom, this is how we would join two amino acids together, amino acid 1 and amino acid 2, and it would form a dipeptide. And a dipeptide is when we have two amino acids joined together, and that has a peptide link around it. And it's hydrolysis because we have this water molecule released here, and we have that dipeptide forming here, and this is our peptide link that forms, and we have that extra water molecule there. Again, hydrolysis, we put that water back in, it would break apart. When we look at the dipeptide in detail, the dipeptide still has that NH2 group at one end, which you can see here, and a COOH group at the other. So if you want to add more amino acids to the chain, you can. You just keep repeating that condensation reaction again and again and again and again, and the chain can get longer and longer and longer. And like I said, that protein can then be broken back down into its individual amino acids via hydrolysis. But you need really, really harsh conditions for this to happen. It doesn't just happen very easily and naturally. So what you would need to do this is you would need to add hot aqueous 6 mole of hydrochloric acid, and then heat the mixture under reflux for 24 hours. And because proteins are condensation polymers, you are able to work out which amino acids a protein chain was made from. And you can use the same method as what we talked about when we were looking at condensation polymers. So if you're not sure about working out that, have a look back at the condensation polymers video that we did before. All you do is you just break each of the peptide links down the middle, and then you either add a hydrogen atom or an OH group to each of the broken ends to get the amino acid back. So for example, with this one, we'd add the OH group to amino acid 1, and we add the hydrogen to amino acid 2. Again, if this isn't really ringing any bells, go back and have a watch of the condensation polymers video. So what we're going to have a look at now is protein structures. So proteins are really big, complicated molecules. And they're actually easy to explain if we break them down into their 4 levels. These levels are called the primary, secondary, tertiary, and quaternary structures. For biology A level, you need to know all 4. But for chemistry, you only need to know the primary, secondary, and tertiary. So we're just going to have a quick overview of those 4 now. So the protein structure, the primary structure. So the primary structure is the sequence of amino acids in the long chain that make up the protein, so the polypeptide chains. And so here's an example of one. And we've got all of our amino acids that make up this group. There's loads more that haven't been drawn on. And that is our primary structure, just that sequence of amino acids that are in that polypeptide chain. Then we have our secondary structure. So those peptide links that form between each amino acid, they can form hydrogen bonds with each other. And that causes the chain not to be straight. It's not in a straight line. And that shape that the chain then takes on is called its secondary structure. And the most common secondary structure is a spiral called the alpha helix. Another common type of secondary structure is a beta pleated sheet. And the beta pleated sheet is a layer of protein folded like a concertina. And then finally is the tertiary structure. So the chain of amino acids is itself often coiled and folded in a characteristic way that identifies the protein. And what can happen is extra bonds can form between different parts of the polypeptide chain, which gives the protein a kind of three-dimensional shape. And this is its tertiary structure. So it can form extra bonds such as disulfide bridges, which we'll have a little look at now. So hydrogen bonds and disulfide bonds, they're great. They actually help keep their proteins in their specific shapes. So that's secondary and tertiary structures we've just looked at. They're formed by intermolecular forces. And what these intermolecular forces do is they cause the amino acid chains to fold or twist. And these intermolecular forces are really, really important. And that's because that three-dimensional shape of the protein is vital to how it functions. So that shape is really, really important for its functionality, how it works, especially if it's going to become an enzyme. So we need its shape to be held really, really strongly and securely. So if we think about enzymes, if we change the shape of an enzyme, it can stop working completely. So there's two main types of bonds that help hold proteins in their shape, and they're hydrogen bonds and disulfide bonds. So hydrogen bonds is one of the type of force that holds the protein in shape. Hydrogen bonds actually exist between polar groups. So for example, the OH group here, and you might have it between an NH2 group as well. They stabilize both the secondary and the tertiary structure of the protein. The amino acid cysteine contains a thiol group, and this is where we're going to look at disulfide bridges. Now, cysteine is really important. So, like I just said, the amino acid cysteine holds a thiol group. And a thiol group is this, SH. Thiol groups on different cysteine residues can lose their hydrogen atom, and then they can join together. And when this happens, they form a disulfide or sulfur-sulfur bond. So here, so they've both lost those hydrogens. So the hydrogens have been lost and they form this disulfide bridge. These disulfide bonds link together different parts of the protein chain and help stabilize the tertiary structure. So hydrogen bonds are secondary and tertiary. Disulfide is just tertiary. Factors such as temperature and pH can affect hydrogen bonding and the formation of disulfide bonds, and can actually cause the shape of the protein to change. So now we're just going to have a quick overview of enzymes, what an enzyme is and how they work. So enzymes are proteins, and these act as a biological catalyst. What that means is enzymes speed up chemical reactions, and they act as a biological catalyst. Enzymes catalyze every metabolic reaction in the bodies of living organisms, and they are proteins. Some enzymes also have some non-protein components to them, but their main body is protein. And every single enzyme has an area called its active site. And this is the part that the substrate fits. The substrate is whatever's going to be reacting, or whatever's going to be taking part in this reaction that needs to be sped up. So the active site, this is where the substrate fits into it so that it can react with the enzyme. And the active site is three-dimensional and makes up part of that tertiary structure of the enzyme protein. And enzymes have a really, really high specificity. They only work with specific substrates, usually one. So one enzyme works with one substrate, another with another. And this is because for enzymes to work, the substrate has to fit into the active site. If the substrate shape doesn't match the active site shape, the reaction won't be catalyzed. And this is called the lock and key model, which you can see at the bottom. So this is the lock and key model. Got a specific substrate that fits the active site. They join together to form an enzyme substrate complex, and then the products are produced. So in this instance, that substrate is going to be split. The enzyme is unchanged after reaction can be reused, and we've formed our products. It can also be that you have the substrates going in and they can bond together as well. It can work either way. And the idea it's called lock and key is because the substrate fits into the enzyme the same way a key fits into a lock. Enzymes are made up of amino acids, so they contain a chiral center. What this means is that their active sites are stereospecific. They'll only work with one N-tiomer of a substrate. The other N-tiomer won't fit properly in the active site. So the enzyme can't work on it. So it's only one specific one that works. Inhibitors are something else that we're going to have a little look at now. So inhibitors slow down the rate of reactions. Molecules could have a very similar shape to substrates. And if a molecule comes along that has a similar shape to a substrate, it acts as an enzyme inhibitor. So it has to have a similar shape to the substrate. And what they do is they compete with the substrate to bond to the active site. And when they bond to the active site, no reaction follows. They block the active site, so no substrate can fit in. So as you can see here, this inhibitor is a very similar shape to the substrate, but we've got this extra bit there. This extra bit here doesn't actually affect it fitting into the active site. It fits into the active site, it blocks it, and that means the substrate now can't fit into the enzyme. How much inhibition happens depends on the relative concentration of inhibitor and substrate. If there's a lot more of the inhibitor, it will take up most of the active sites, and very little substrate will be able to get to the enzyme. And the amount of inhibition is also affected by how strongly the inhibitor bonds to the active site. And finally, some drugs work as inhibitors. So some drugs are inhibitors, and what they do is they'll block the active site of an enzyme and they will stop it from working. So for example, some antibiotics work by blocking the active site of an enzyme in bacteria that helps make their cell walls. This causes their cell walls to weaken over time, so the bacteria eventually burst. The active site of an enzyme is very, very specific, so it takes a lot of effort to find a drug molecule that will fit into the active site. It's even trickier if the drug molecule is chiral. Then only one N-tiomer will fit into the active site because the active sites of enzymes are stereospecific. So often these new drug molecules are found by trial and error. Scientists will carry out experiments using lots of compounds to see if they work as inhibitors for a particular enzyme. They'll then adapt any that work and try and improve them. And this process takes a very long time. One way scientists are speeding this process up is by using computers to model the shape of an enzyme's active site and predict how well potential drug molecules will interact with it. By doing this, they're able to quickly examine hundreds of molecules to look for ones that might be the right shape before they start synthesizing and testing things in the laboratory. And we're just now going to have a look at a couple of exam questions based off proteins and enzymes. So the structures of amino acids glycine and serine are shown. When two amino acids react together, a dipeptide is formed. And you're asked to draw the structures of the two possible dipeptides that could be formed when serine and glycine react together. So what's really important here is the word two. There's two possible dipeptides that you could form, and you're asked to draw both of them. Doesn't matter which one you draw first, you just need to make sure you've drawn both of them. So the one I'm going to draw first is going to be this one with serine at the front. I'm going to have H2N, my carbon here. I'm going to have CH2 and my OH here. As you can see, that's my serine that I'm starting to draw here. Hydrogen here. Carbon. Double bond to my oxygen, and we're going to get rid of that OH group off. Then it's going to bond with the nitrogen from my glycine. We're going to get rid of one of those hydrogens off there. And it's going to bond to carbon, hydrogen, hydrogen, OH. Other one we can draw is we could have H2N, H, H, C. And so these are the two structures of the possible dipeptides. So as you can see, it's just a difference of which way around that dipeptide link has formed. So it's basically if it was glycine plus serine, or if it was serine plus glycine. So this one's if serine then joined to glycine, and the other one's the opposite way around. Question 2. Sometimes adding a chemical to an enzyme catalyzed reaction can cause the enzyme to stop working properly. You're asked what are chemicals that prevent enzymes from working called. All you need to do for that is just write down inhibitors. Then part B, you're asked explain how a drug molecule may prevent an enzyme from working properly. So here you want to say something along the lines of a drug molecule may have a very similar shape to the enzyme substrate. And fit into its active site. That gives you the first mark. Now, so I want to point out here, I said a very similar shape. I've not said the same because they are not identically the same. Otherwise, it would just be the substrate. So the drug molecule may have a very similar shape to the enzyme substrate and fit into its active site. Then explain how it stops the enzyme from working properly. This prevents, this blocks even. This blocks the active site, preventing the substrate from entering. And then that gets the second part of that mark. And then final part of this question, a scientist has developed a drug that stops a bacterial enzyme from working. The drug molecule is chiral. Explain why only one entamer of the drug will be effective against the enzyme. This is the really important part to start off with marks, is this key fact. The active site of an enzyme is stereospecific. First mark. So only one entamer will be able to fit into the active site. Able to fit into the active of the enzyme. And that'll get you the second part of that mark. Okay, and that was our session on proteins and enzymes.